Okay, so we're going to start with a brief review of what we are going to be talking about in the whole course, meaning different types of microbes, their structure, which branches of microbiology study them. So, microorganisms. We broadly divide them into several categories. Category number one, bacteria and Archaea. As you will see soon, it is a very artificial grouping, but these two types of microorganisms, what's common? They all are prokaryotic, which means they do not have nucleus or membrane-bound intracellular organelles, and they always unicellular. So, immediate question. They are always unicellular. Why? The short answer is we have no idea. The modern science suggests that most likely they are always unicellular because they lack sufficient communication between the cells. Now, question mark means that we don't really know if it's true, okay? So the branch of microbiology that studies bacteria and archaea is bacteriology, not a rocket science. Next group are fungi. So fungi are eukaryotic. They can be unicellular. We call such fungi yeasts, or multicellular. We call such fungi molds. And the branch of microbiology studying these microorganisms is mycology. Then we have Protozoa. Now I want to focus your attention, it is not a taxonomy. So it's not like genus or phyla or classes, it's just breakdown on the structural, by the structural features. Protozoa, by definition, are unicellular eukaryotes. So we just place all unicellular eukaryotes, well, except some, into the same group, okay? Protozoa usually fall in the realm of parasitology studies, as well as helminths. Helminths are the second best microorganisms. They are eukaryotic, multicellular, always multicellular organisms. So that's actually the only always multicellular organisms that we're going to be looking at, also studied by parasitology. Now we have two more. We have viruses and algae. I'm going to put algae first, and that's, I believe, penultimate time we talk about algae. We're going to talk about them one more time. So algae are eukaryotic. It can be multicellular or unicellular. And they are studied by the branch called phycology. Now these ones, viruses, are absolutely the best microorganisms, and these are acellular. Viruses do not have a cellular structure, which means they are non-living. viruses are not considered to be living things. Studied by virology. Now, the name of the subject that we are working on in this class is microbiology, right? Which means small. So how small? To answer this question, we need to highlight whether we can look at microbes using light microscopy or electron microscopy. I'm going to be saying a phrase, in most cases, a lot. Basically, there are a lot of exceptions in microbiology. So, bacteria and archaea, in most cases, light microscopy is enough. Fungi, well, practically in all cases, light microscopy is enough. Protozoa, definitely light microscopy is enough. Helminths, you can look at them with a naked eye. In overwhelming majority of cases, however, I can think of at least one schistosoma that you would need a microscope to look at, but light microscope. Algae, naked eye, obviously, or light microscopy. And finally, viruses. So viruses, overwhelming majority of them, you can see only using electron microscope. Now, again, majority of cases. So if I ask you a question, can we see some viruses using light microscopy? Yes. Can we see actually some bacteria with a naked eye? Surprisingly, yes. There's like a couple of species that are large enough, tiny, tiny, tiny little ones, but you can see them. Does that make sense? So you should be able to answer the question to characterize, like, which of the following characterizes helminths or protozoa or bacteria or fungi. Okay. Without any sort of further ado, let's talk immediately about the taxonomy and how we classify microorganisms. So first of all, the classification. What do we base it upon? There are two classification systems. The first one was introduced by Whitaker. So Whitaker classification groups microorganisms based on the appearance, the ecological niches, basically phenotypical similarities, okay? And you probably saw this in the lecture notes. There's a little tree with Monera, Protista, Fungi, Plantae, and Animalia. Okay, does that make sense? So these are 5 kingdoms. So Whitaker included Monera, all prokaryotic organisms, okay, in one kingdom, all unicellular eukaryotes in another kingdom, Protista, placed all fungi right here, yeasts are not here, okay, and then plants and animals, kind of obvious thing, right? So, and it worked pretty well because you have to understand taxonomy is an artificial science. I mean, it's not a science, it's just artificial product. You know, you can organize your cloth based on the size, based on the length, based on the color, I don't know, material, whatever. So another scientist, Carl Woese, was working on genotyping bacteria, and to make a long story short, he came to an experimental observation and a conclusion that we have to reconsider the classification. So Woese-Fox system, that is the main system accepted in the biology now, is based not on the phenotypical similarities, but on the similarities between the 16S RNA gene. the premise that Carl Woese was operating on was as follows. Closely related, so he said that there are some very, very conserved genes, genes that are not changing a lot. Does that make sense? And the organisms that are closer to each other will have those genes much more similar. So for instance, our 16S RNA gene and human primates 16S RNA gene, okay, will have an enormous amount of similarity, but if you compare ours to mice, similarity will be much less. You following me? So this similarity suggests that two organisms are evolutionary closer and are in a closer relationship, genetic relationship. So based on these observations, Woese changed, reconsidered this whole classification scheme, saying that there was a last universal common ancestor. I know there's a cartoon now streaming somewhere on TV. And then it produced bacteria, okay? And the second branch actually split into Archaea and Eukarya. So those are three domains of life, okay? Does that make sense? So basically, the idea that Carl Woese had was that the initial last universal common ancestor evolved, producing a group, a domain of bacteria, domain of Archaea, and domain of Eukarya. And if you look at this, what's called phylogenetic tree, you can see that Archaea and Eukarya are closer to each other. Does that make sense? While bacteria is farther from them. It's kind of a bizarre because under the microscope, let's say, bacteria and Archaea look very, very similar. Well, you can imagine Archaea that are prokaryotic are structurally very different from eukaryotic organisms. It's basically the same situation when you have a brother and a sister, and they have a cousin. And brother and a sister don't look alike. However, two cousins look very similar, but genetically they are more distant. Is that clear? That make sense? Okay, now, how do we name organisms? We use binomial nomenclature, where you have name one and then name two, okay? So name one designates the genus. And both name one and name two designates species. So for instance, Staphylococcus aureus belongs to the genus Staphylococcus, represents the species Staphylococcus aureus. Bacillus subtilis belongs to the genus Bacillus, as a species Bacillus subtilis. Does that make sense? Questions on this one? Viruses fall into their own category. Both the taxonomy of viruses is very kind of a shit show. Kind of complicated because they're not living, there are no cells, so it's really hard to assign species versus genera versus families and so on. People try, as well as naming. Naming is another big problem. Any questions on this part? No, we good? Okay, awesome.